This session is the continuation of our on-site lecture on Introduction to Medical Microbiology. Last time we covered the principles of microbial pathogenesis. We talked about some historical developments in the field, and we also covered concepts on epidemiology and control and management of infections caused by different microbial pathogens. As a review, we divide the science of microbiology into two. So there are basic microbiological sciences that relate to the microbe as the unit of analysis. Okay, so primarily we are interested in the microbe's identity under taxonomy. We study microbes from different groups. There are dedicated fields to study them. There's a field to study bacteria, um, cyanobacteria or algae, fungi, protozoans, parasites in general, and viruses. Now, looking into the characteristics of the microbes, we are also interested in what's happening inside them, primarily dictated by the genetic material or the genome of these microbial agents. So we are interested in how they process energy, how they extract energy from the environment, how they produce energy, and what distribution patterns they demonstrate in the environment or in the different reservoirs where we find them. Okay, how distributed they are. Okay. Also, in relation to disease, we covered or we study how these microbes evade the immune system of the host. We study their normal distribution as infectious agents in the human population, and where do they come from, how they are transmitted from their source to hosts, in general the life cycle of these microbial agents. Okay. Now, once we know more about, or once we've established a basic understanding of these microbes, we may be able to apply such information or such understanding to different aspects or different fields. In the industry, some microbes are harnessed as biotechnological factories for the production of fine chemicals, um, for the production of medicines, for example, and also in the food industry, very important. Since specific microbes facilitate food processes or processes that relate to food production. Okay. Environmental microbiology is a field that covers the study of microbes in different ecosystems, as we relate also these ecosystems as reservoirs for these microbes in that essentially is a key aspect in their life cycle. Okay, in terms of control or controlling their presence or growth, we have the study of infection biology and also treatment. So for medical microbiology, it's a hybrid of these two things. So moving forward, all our discussions about microbial pathogens, representative pathogens from each group, we will be talking about this five important things to further appreciate their significance as human pathogens. Okay, so in a nutshell, medical microbiology covers the study of microbes that affect human health. More importantly, as emerging pathogens are identified, we recognize the importance of interactions between the microbe and other carriers or other hosts such as non-human animals. Okay, since we've seen that, or we've through the years we've identified microbial agents from viruses, bacteria, fungi that are originally adapted to live or adapted to infect non-human animals, but later on were transferred or had the capacity to also invade human hosts. So as a review, these are the kinds of pathogens we'll cover all throughout the semester. So for the midterms, we'll cover primarily the classic bacteria, viruses, and fungi. For the finals, we'll cover not only microbial agents, microbial parasites such as protozoans, but we'll also cover multicellular parasites as well as invertebrate animals that can cause medically significant human infections. Okay, so just to emphasize a few things on the nature, the biology of prokaryotic and eukaryotic microorganisms. We reviewed that these microbes or prokaryotes in general have a very different genetic makeup. So most bacteria or all bacteria are known to have a circular DNA molecule, unlike eukaryotes that are more complicated, and there are multiple chromosomes that are involved, unlike in prokaryotes. The localization of DNA for prokaryotes is not within a membrane-bound compartment inside the cell, unlike in eukaryotes. More so, um, there's absence of intercellular organelles such as mitochondria, the endoplasmic channels, for the case of prokaryotes, unlike in eukaryotes. In terms of reproduction, okay, prokaryotes mostly reproduce asexually by binary fission, while eukaryotes, in most cases, undergo sexual reproduction, and sometimes, or in some species, are able to reproduce asexually. Okay. These terminologies, or most of these, I've already discussed from the start or since last week. I just have to cover a few things. So, opportunist or facultatively pathogenic microorganisms are those that can cause infection when their environment or the context allows them to. So in our case, as we have normal microflora in our body, in the different parts of the human body, and in instances where the immune system is down, or there's a drastic change in the normal factors that dictate the everyday condition of our bodily organs or bodily systems. That's an instance when some normal microflora can actually cause infection. Okay. I also talked about subclinical infection or asymptomatic cases. So, technically the host is infected, but there's no manifestation of any sort in terms of the symptoms that are associated to the infection cause. Also, I'd like to cover two other terms. We have anthropognosis and zoonosis. So when we say that an infection is an anthropognosis, that means that it is transferred from one person to another, one human to another. The chain of infection is said to be homologous, while zoonosis typically are used as is a term used to describe transmission of an infectious agent from other vertebrates, okay, non-human animals to humans, okay. and occasionally vice versa. The chain of infection is said to be heterologous, as participating hosts are of different groups or different types. No? One human and one non-human. Okay. As medical microbiologists, we study the following things about microbial pathogens: their physical chemical characteristics, how they grow, how they are transmitted, okay, from the environment to the host, or generally we look at their life cycle, okay, and how they cause infection. So here we will cover discussion on the immunology of the host and the immune evasive mechanisms of the pathogen. Okay. So a good way to remember the nature of microbial pathogens we cover in the study of medical microbiology. So they are microscopic, independent units that more often than not are really complex living organisms. They have rapid growth rates. They can grow at most of them. Okay, I'm not saying that all of them, but most of them grow rapidly to adapt to the host, and they are most of the time present everywhere. They're omnipresent. Okay, so now that we've talked about some of the intro principles of medical microbiology, in addition to the study of pathogenesis and transmission, okay, medical microbiology also covers the study of treatment and management of the infection. Okay, so for the case of bacteria, bacteriological management of an infection in a patient usually involves the following steps. So there is the establishment of a clinical diagnosis, a definitive clinical diagnosis. And to prove that, okay, we isolate the causative agent, following the germ theory of infection, that there should be an infectious agent. But of course, we have to remember that there are limitations to the classical theory. We talked about the importance of molecular markers as additional factors we consider to diagnose or to really establish the presence of an infectious agent. Also, more recently, it's important, or part of the workflow in the management and treatment of infectious agents, is the identification or the establishment of whether they are controllable with specific drugs available. Okay, so we establish their antibiotic susceptibility so that we can identify which drug, which medicine to use to control them, to manage them. That as the um, medico, as the medico applies therapeutic management, that management is monitored so that we can see how the body responds with an aid, okay, in the presence of drug aid that is intended to kill or intended to eliminate the microbial pathogen. And we confirm, as symptoms weighing down, we confirm if the microbial pathogen for the case, or the bacteria for this in this discussion, in this example, we confirm if it's really cleared, no, if there is the bacteria is no longer present in the host. Okay. Um, so at the broader context, we also look at the epidemiology of microbial pathogens, what carriers or vectors are involved in the transmission, the life cycle of such pathogens. And we also recognize different vehicles of infection, different routes of transmission, whether animate or inanimate matter are involved in the process, no, or in their life cycle and transmission mechanisms. At the looking at the epidemiology lens, or using an epidemiology lens, okay, community infection is definitely um, is definitely a concept that will be recurring as we discuss microbial pathogens, especially those with wide distribution and those that are known to cause community acquired infection, or known to cause community acquired infections. Okay, so important that we have precise data, we are able to identify the source and the process of spread. Um, Also, we are important that we establish whether the microbial pathogen is actually a zoonotic one, that if it originated from a non-human host that was able, and it was later on able to adapt to the human body. Okay. Right. So the last section or the last slides for my presentation will cover different microorganism groups associated with our body. And we divide them typically into two. There is transient flora and there is resident flora. Okay, so let's discuss each of them. So first, transient flora, as the name suggests, actually includes microflora that are usually derived from the surroundings, from the environment. They do not really stay in our tissues permanently. So transient means temporary. Okay, so they only stay for hours, days, or weeks, but they do not maintain an intimate relationship with the human body, with human tissues. Okay, generally, they're not significant as they are as they come and go, and in the presence of an intact microflora, they should not be able to cause any serious infection, any serious sequelae. Okay, because their nature is they colonize our tissues for a specific time period, but then they go away. They don't really stay. Okay. On the other hand, resident or normal microflora of our body are the ones that we typically identify as commensals. So they stay in human tissues for longer periods of time or all throughout the lifetime of an individual, and they are usually associated with our skin, our mucous membranes, no, and they have developed this intimate relationship with the animal body, with the human body. In instances where their normal population structure is disturbed, like if you're taking an oral antibiotic, no, your gastrointestinal tract, which contains resident or normal microflora, they can also be targeted by such antibiotics. So they can reestablish themselves in such instances where when their population structure has been modified because of the use of antibiotics. Okay? Normally, they are harmless since they have this intimate relationship with us. They don't really cause the usual infections. No? But in the absence of also in the absence of other ano no you coincidental abnormalities, or we can term as predisposing factors, they are part of our body. They don't cause infection. Okay, so a good example or discussion on that is um, as long as the host possesses a um, healthy immune system, or an immune system that is not compromised, these resident microflora are usually harmless. They may cause diseases, however, in instances when they are introduced to body parts that they don't usually thrive in. So, what are these parts? The blood, for example, the more systemic structures, or the more important structures like the lymph, the nervous system. So as long as these normal microflora are absent in those important uh, uh, areas of the human body, these microflora will not cause infection. Okay. So some examples of resident or normal microflora in our body, there are certain parts or specific um, tissue groups or organ systems with associated normal microflora. There are microflora in our skin, okay, which mostly are bacteria, fungi, and such as yeasts. There are also microflora in our oral cavity, in our mouth, inhabiting our teeth, such as Streptococcus bacteria, Streptococcus genus. There are also some representative from fungi, such as Candida. Okay. There are also groups such as the usual bacteroids and actinomyces. Okay. In our throat, almost the same group or similar nature or categories of normal microflora, Streptococcus, the Streptococci group. Okay. There are also others that are belonging under Staphylococcus genus and Haemophilus. Okay. In our nose, okay, almost the same microflora, right? Dipteroids, take note, can inhabit our nasal cavity along with strep and staph. Okay. Now, going inside internally in the gastrointestinal tract, especially in the intestines, these are the known groups of bacteria that are considered normal microflora found in our intestines, mostly lactobacilli, enterobacters, okay, bifidobacterium, clostridium, um, what else? In instances, some Klebsiella genus can also be found in the intestines. Okay. For the reproductive system, okay, for especially for the female human, there are resident normal microflora in the urethra and vagina, as shown below. Okay. Um, Now, okay, all right. So some more notes on the presence of these normal microflora per organ system or tissue group. So in our skin, most of these microbes, most of the resident microflora are found in our hair follicles or in our sweat glands. So they inhabit our hair follicles or sweat glands. So for the case of Cutibacterium, that may cause infection in the manifestation of pimples. Okay, they're restricted to, for as an example, that species is restricted to our hair follicles. In our throat, okay, in instances when these groups of microbes become opportunistic or facultatively pathogenic, they can cause upper respiratory tract infections that are manifested by symptoms such as cough, itchiness, throat dryness. Sometimes can cause or most of the time can cause fever, as your body will respond to controlling the growth, controlling the population of these throat inhabiting normal microflora that later on become or in some instances became facultatively pathogenic. In the vagina, we have lactobacillus and candida, lactic acid bacteria and yeast that are usually inhabiting the female reproductive tract, and these species or genus they thrive in a specific pH. So a change in the pH of the female reproductive tract can enhance the or can um, change the pathogenicity or the not really pathogenicity but can change the can send signals to these genus of microbes for them to become more invasive, no, and not normal than usual, right? Then in the urethra, bacteria, these representative normal microflora can also cause urinary tract infections when they grow out of they grow out of proportion in terms of their number. Okay, I will stop now. I'll cut the recording into two.